Welcome back to the deep dive. So you've handed us this incredible trove of scientific source material.
It's quite a stack.
It really is. We're talking articles, deep research papers, and they all seem to offer this direct molecular comparison between two, well, two very different philosophies in vaccine design.
Right. On one hand, you have the Western mRNA platforms.
Pfizer and Moderna.
Exactly. And on the other, you have the designs that are linked to General Zoo and the PLA Academy of Military Medical Sciences.
And their focus was more on subunit and vector platforms.
It's a spectacular set of sources because it really forces us to do a true comparative analysis. We're not just, summarizing clinical trials. We are getting into the subcellular engineering choices.
So what's our mission today?
I think our mission is a granular comparison of these two approaches. And we have to look at it through two very specific interconnected lenses. First, Type I Interferon suppression.
So IFNI, how they sneak the antigen in.
Precisely. And second, the resulting risk of what's called molecular mimicry and autoimmunity.
And this really gets to the core paradox of, well, all of modern biological engineering, doesn't it?
It really does.
I mean, for a vaccine to be highly effective, it has to deliver its message, the genetic code or the protein payload, really robustly. But our bodies have spent, a millennia evolving to instantly detect any foreign genetic material.
Or even just high concentrations of a viral protein.
And that first line of defense, that's the innate immune system, right? And its most potent alarm signal is that IFNI.
That's the tightrope walk. It's a perfect analogy. You have to deliver the antigen effectively.
No antigen, no response.
Exactly. And that means you have to bypass, or in some cases, actively suppress the innate immune system's detection.
Okay.
And the sources show that the molecular mechanisms these two platforms used to do that are, they're fundamentally different. One is chemical cloaking, the other is more of a functional antagonism.
But the irony, as you put it, is that they might lead to a shared vulnerability.
A shared pathological vulnerability. That's the profound part.
That sets the stage perfectly. So let's jump in, part ein, and let's start with the tech that most of our listeners are probably familiar with, the Western mRNA platform.
BNT162b2 from Pfizer and mRNA 1273 from Moderna.
How did they solve that first big problem, silencing that innate alarm?
Well, the story of the Western platform is, it's really a triumph of chemical engineering. You have to think of the innate immune system as like the cell's internal security guard.
Okay.
If you inject natural exogenous RNA into the body RNA that isn't properly capped or tagged as self, that security guard sees it instantly. It sees it as a pathogen trying to hijack the cell's machinery.
And what does the security guard do when it sees the intruder?
It pulls a fire alarm, a massive alarm response, which is the Type I interferons, IFNI.
And that's a whole cascade of signals.
It's a huge signaling cascade.
Yeah.
The natural RNA gets picked up by sensors in the endosomes, like toll-like receptors 7 and 8.
TLR 7 and 8.
And by sensors just floating in the cell fluid, the cytosol. Those are primarily RAI and MDA5.
So if any of those get tripped,
the cell goes into total lockdown, a complete antiviral state.
Which means it stops all production, starts shredding any foreign RNA it can find, and basically sends out a warning to all its neighbors.
Right. Prepare for an attack.
The vaccine fail?
It fails instantly. A robust IFN response wouldn't just sound the alarm, it would trigger the destruction of the vaccine mRNA. The whole thing gets chewed up before that crucial translation step, the part that actually makes the spike protein.
So if the vaccine is detected, it's destroyed. Game over.
Game over. The whole challenge was just getting past that initial detection.
So the solution had to be molecular camouflage, the now famous chemical cloak. Tell us about that.
This is the genius of N1-methylpseudouridine.
I'm the one.
M1A, exactly. This single modification is really the key piece of intellectual property that unlocked the whole thing. So, RNA has 4 letters, A, U, C, G.
Right.
Instead of using natural uridine, the U, the manufacturers just replace all of it with this M1. It's a tiny, tiny chemical tweak to the uridine molecule structure.
How does such a small change fool a defense system that's been evolving for millions of years?
The sources explain it really well.
Yeah.
The M1 modification essentially makes the RNA invisible to those innate sensors.
It cloaks it.
It cloaks it. The TLRs, RIGI, MDA5, they all evolved to recognize the specific molecular patterns of foreign unmodified RNA. When you add that methyl group and change the structure to pseudouridine, you shift the recognition profile just enough.
So the sensors either think it's self or they just don't see it at all.
They just fail to recognize it as a threat. It's a remarkable invisibility suit. And by preventing that initial recognition, They suppress the entire IFNI signaling cascade.
Yeah.
They achieve the bypass.
And that was the intended goal. A massive success.
A massive success for engineering, yes. This chemical silence allows for, well, prolonged stability and really robust, high-quantity translation of the mRNA into the spike protein.
So the antigen gets produced for way longer than natural RNA ever would.
Much longer. And that leads to this huge initial immune response.
But, you said it before, a successful bypass has downstream consequences. The sources are pretty clear on this. When you silence that innate warning, what does that do to the quality of the immune response that comes later?
This is a really critical point. We need to unpack it carefully. So the adaptive immune response, your T cells, your B cells, when that's generated without the initial strong inflammatory signal from a big IFNI storm,
The character of the immunity can change.
it can change. IFNI isn't just an alarm bell. It heavily influences what's called immune polarization. It pushes the response towards a strong inflammatory Th1 type.
Which is what you want for clearing viruses.
It's crucial for clearing viral infections.
So without that Th1 push...
The system can.
Yeah.
It can skew. It can move towards a less inflammatory, more regulatory, or even a tolerizing response.
Tolerizing, meaning it learns to ignore it.
Exactly. The sources suggest this regulatory bypass can help the antigen persist, especially if you get repeat doses. Over time, the immune system might start to treat the spike protein less like a threat to be violently eliminated and more like, I don't know, a persistent, non-harmful environmental thing like pollen.
And that brings us to what you mentioned before, immune tolerance and these IgG4 antibodies.
Precisely. The generation of IgG4 antibodies is a molecular marker. It's often associated with chronic, non-inflammatory exposure or immune tolerance. It's the kind of response you see in, like, successful allergy desensitization shots.
Interesting.
So the sources suggest that repeated dosing in this IFNI-suppressed environment might favor making these non-inflammatory IgG4 antibodies, and that would indicate a state of immune acceptance or tolerance.
Not the sterilizing, neutralizing attack you need to completely clear a virus.
It's a profound, very subtle trade-off. You're maximizing the initial yield of the antigen, but you might be regulating the long-term character of the immunity.
So the chemical change solves the delivery problem, but maybe introduces a regulatory problem later. Okay, let's pivot from the delivery system to the payload itself. We have to talk about the structure-based antigen design, the famous S2P lock.
The S2P lock is another just foundational piece of engineering brilliance. You have to remember, the goal wasn't just to make a spike protein. It was to make the spike protein in its most stable, most functional, and most immunogenic form.
And that form is the perfusion conformation.
The perfusion shape. The shape it holds right before it snaps open and fuses with our cell membranes.
Why is that perfusion state so important for making antibodies?
Because it exposes the most critical neutralizing epitopes. Those are the parts the B cells absolutely have to see to generate effective antibodies. The problem is, the spike protein, it's floppy. It's structurally unstable.
It likes to snap into the post-fusion state.
Spontaneously. And if the vaccine made post-fusion spikes, the antibodies you'd get would be, well, much less effective.
So how did they lock this notoriously floppy structure into that perfect perfusion shape?
This is the S2P patent. It's a very, very precise molecular surgery. They substitute two specific amino acids, K986 and V987, with prolines.
Prolines? Why prolines?
Proline is a unique amino acid. It's rigid. It introduces this kink or a lock into the protein chain. So by putting two prolines right at the spot where the structure naturally wants to snap.
They act like molecular staples, a structural lock.
That's a great way to put it. They're forcing a conformation that nature didn't really favor in isolation, but one that is optimal for a vaccine.
And that guarantees the spike protein stays stable in that perfusion shape.
And that yields an incredibly potent and, importantly, a focused immune response against the right targets. And that focus, as we're about to see, is both the strength and the ultimate vulnerability of this design.
Before we get to the vulnerability, let's just quickly touch on the origins here. The sources highlight the IP trail for the S2P lock.
Yes, the sources identify the key intellectual architects. That's Dr. Barney Graham, Dr. Kizzmekia Corbett, and Dr. Jason McClellan. And it's important to note, the patent for this S2P technology itself, it's held by the U.S. government, the NIH and UT Austin.
So this technology is platform agnostic.
Completely. It's about the structure of the antigen. It was widely used across many different vaccine candidates globally. This isn't just a scientific finding. It's a critical piece of biomedical infrastructure.
Okay. So we've covered the engineering success. Delivery system is cloaked with M1A, payload is locked with S2P. Now we have to transition to part two. This is the convergence point, the shared risk. And it's not from the delivery system, but from the spike protein itself.
This is where the complexity of molecular biology it reveals itself. The risk centers on the specific tiny 5-amino acid sequence,
Yeah.
the pentapeptide RLFRK.
RLFRK. Arginine, leucine, phenylalanine, arginine, lysine. That seems like a lot of trouble packed into 5 letters. Where is it?
It's located in the most critical real estate of the entire spike protein, the receptor binding motif, or RBM.
The RBM. So that's the exact loop that makes first contact with the ACE2 receptor on our cells.
The very tip of the spear. And because it's right there on the surface, it's highly exposed. And, critically, the sources cite experimental evidence that this exact sequence, or fragments containing it, have been detected in the plasma of vaccinated people.
Detected in the bloodstream.
Yes.
And pretty quickly, too, from what I remember.
Incredibly fast. The detection window is as early as 1 day post-dose 1 for Moderna. For Pfizer, it's within 15 days post-dose 1. This fact is vital.
Why?
Because it means the peptide isn't hidden away. It's being shed, it's exposed, it's detectable in the blood. That makes it readily available to interact with T cells and potentially trigger an immune response that mistakenly targets our own tissues.
The essence of molecular mimicry.
That's it.
Okay, let's spend some real time on the first mimicry scenario because it's pretty alarming. The potential for, as the sources put it, ablating the IFN system.
We're coming full circle back to the failure of innate immunity.
Yep.
So the first scenario connects this tiny viral sequence directly to our body's ability to produce its core antiviral defense. The target here is a human protein called PPIP5K2.
TPIP5K2.
And the viral RLFRK sequence shares what's called homology, a structural similarity with a part of that human protein, specifically the sequence RLFRK high.
So they look alike. But similarity doesn't always mean trouble. Why is PPIP5K2 so important?
It's not some random protein. It is absolutely essential. We need a clear analogy here to really grasp its function. Think of the entire IFN production pathway as a car engine.
Okay, an engine.
The ignition switch for that engine is a protein called IRF3, interferon regulatory factor 3. It's the master regulator. Once it's activated, it rushes into the nucleus and flips the switch to mass-produce interferon.
The central switch.
Right. But for IRF3 to activate, it needs to be phosphorylated. It needs a spark to turn the key.
And where does the spark come from?
That spark is a high-energy molecule called 1IP7. But 1IP7 has to be made, the factory that synthesizes it. That's the job of the kinase we just named. PPIP5K2.
Okay, let me see if I have this. PPIP5K2 is the generator that makes the high-octane fuel, 1IP7. And that fuel is needed to power the ignition switch, IRF3, which starts the entire antiviral engine, IFNI.
That's the perfect analogy. And PPIP5K2 is the bottleneck. 1IP7 is strictly required for IRF3 to be phosphorylated. So PPIP5K2 controls the entire process.
All right, now let's close the loop with RLFRK. If the vaccine makes T cells that recognize that viral sequence, how does that hurt our own machinery?
Well, if your immune system, specifically these cross-reactive T cells, which are often linked to certain HLA types like HLA-DRB108.01, if those T cells are sensitized against the viral RLFRK sequence,
they might see our own PPIP5K2 and think it's the enemy.
They may mistakenly recognize and launch an attack against the homologous human protein.
Against the generator.
Precisely.
Yeah.
And the resulting damage or inhibition of PPIP5K2 means the host's ability to make that 1IP7 fuel just. It degrades.
No fuel, no spark.
You can't phosphorylate IRF3 effectively. The ignition switch fails to turn.
So the body has launched a friendly fire attack against its own fundamental antiviral defense system.
And this creates what the sources suggest is a vicious cycle.
Which is?
Autoimmunity leading to acquired immunodeficiency. The consequence is a biological failure of the innate immune response. And that leaves the host systemically vulnerable.
Not just to SARS-CoV-2.
No, potentially to other pathogens, or the persistence of chronic viral infections. It's a pathology where the immune response meant to fight the virus ends up attacking the machinery needed to fight all viruses.
That detailed mechanism that's what makes this material so compelling. But there's a second target for RLFRK, right? This one shifts from immunological to neurological.
This is the second scenario, and it's linked to demyelinating disease. The RLFRK sequence also shares mimicry with another crucial host protein called tenerin-4 or 10NM4.
Tenerin-4, what does that do?
And tenerin-4 is a cell adhesion molecule. It's absolutely critical for the proper development and maintenance of the central nervous system. Its main role involves axon guidance and, most importantly here, CNS myelination.
Myelination, that's the insulation, the fatty sheath around our nerve fibers that let signals travel fast.
Exactly. 10NM4 helps regulate the formation and the integrity of that protective coating. So, if the immune system, sensitized by the RLFRK epitope from the vaccine, generates cross-reactive T cells or antibodies against 10MM4.
You get an autoimmune attack on the nervous system's insulation.
And the clinical link is pretty stark. Autoimmunity against 10MM4 is known to be linked to demyelination and neuroinflammation. The sources suggest this provides a credible molecular explanation, a smoking gun at the molecular level for some of the documented reports of demyelinating conditions seen after vaccination.
Like transverse myelitis or multiple sclerosis?
Yes. It's really staggering that 5 amino acids within the RBM could carry this dual profound threat, collapsing innate immunity and attacking the nervous system.
Wow. Okay. This deep dive is really showing the unintended costs of this kind of antigen engineering. Now, we need to execute a major pivot. Part 3. General Zhu's platforms.
A completely different approach.
His team, with the PLA and AMS, they focused on subunit vaccines, viral vectors. Their approach to IFN suppression was fundamentally different. No hiding. It was functional suppression.
The intellectual contrast is immense. The Western approach solved the problem by hiding the RNA from the sensors, using M1A.
Right.
Zhu's approach was to actively engineer the spike protein to turn off the alarm, leveraging the exact mechanisms the virus itself uses to antagonize our immune systems.
Okay, let's break that down. The first mechanism of active suppression, functional IFN suppression via the RGD motif.
So Zhu's team, in their subunit designs, they either utilized or retained a key functional element, the RDD, or arginine glycine aspartate motif.
That's naturally on the spike protein.
It is. And unlike the Western platforms, which focus so heavily on the S2P lock for stability, Zhu's team often retained elements like RGD. Sometimes the idea was to broaden the antibody response. But that choice came with a powerful functional consequence.
And that consequence is mediated by our own integrins.
Yes. The RGD motif is one of the most famous binding sequences in all of biology. It's a known ligand for a family of cell surface receptors called integrins.
Like alpha, beta, 100 and 101?
Exactly. And they're highly expressed on all sorts of cells. So when the spike protein's RGD motif binds to these host integrins on the cell surface, it kicks off the suppression cascade.
What's the downstream effect of that RGD integrin binding?
That binding is the mechanical trigger for the active release of transforming growth factor beta, TGF-PA.
TGF-PF. I usually associate that with things like cell growth, wound healing.
Scar tissue formation, which is why it's usually kept locked up, sequestered in the extracellular matrix. The RGD interaction helps activate it and release it.
So what's its role here as an immune modulator?
It is a powerhouse immunosuppressor. Once it's released and active, the source is concerned that TGF-beta directly downregulates the expression of Type I interferons.
So it tells the cell, stop the inflammatory response. Switch to a quiet, healing state.
That's exactly its message. So this functional antagonism built into Zhu's design used a natural motif, RGD, to commandeer an endogenous host pathway, TGF-2 release, to actively suppress the antiviral state, creating a pro-viral environment, as the sources call it. It's an intellectual leap from passive camouflage, M1A, to active signal hijacking immunosuppression.
All right, now let's move to their second strategy, immunofocusing, which, kind of like the Western stabilization, reveals a similar paradox of unintended risk.
Immunofocusing was Zhu's rational design strategy. Their goal was to make the vaccine response incredibly efficient by removing distractions.
So they specifically aim to remove non-neutralizing epitopes, often by just cutting out the entire S2 subunit of the spike.
Exactly. The S2 subunit is often highly conserved across coronaviruses, and it generates a large antibody response that it doesn't necessarily help with potent virus neutralization.
So it's a distraction for the immune system.
It can be. By focusing the immune response, they aim to generate only the highest quality neutralizing antibodies.
So they were making a surgeon's scalpel where the Western platforms made a broadsword. They concentrated the immune response entirely on the receptor binding domain, the RBD.
Yes, and that is the paradox. This rational optimization, it inadvertently maximized the risk.
How so?
By intensely concentrating the immune attack on the RBD and its receptor binding motif, the RBM, Zhu's platform was perfectly optimized to generate a highly specific, robust immune attack against the one sequence we know is a critical vulnerability.
RLFRK.
RLFRK.
It's a tragic intersection of rational design and molecular coincidence. They solved the efficacy problem by focusing the attack, but they ended up optimizing the attack against the PPI-P5K2 and 10M4 mimics.
And the sources confirm this intellectual distinction. We have to reiterate, Zhu filed patents early February 2020 on subunit vaccines and viral vectors focused on the RBD. These are mechanistically and structurally distinct from the M1-based mRNA platforms with the S2P lock.
But both paths led to a massive immune response against the RLFRK epitope.
They converge on the same molecular target.
Now, let's drill down into the foundational knowledge that their team had. The sources emphasize their deep understanding of how SARS-CoV-2 naturally dismantles the IFNI response.
This part really reveals the depth of their virological expertise. They were working from a known playbook. They had characterized the specific mechanisms the virus uses to evade IFNI during a natural infection. You can think of it as the virus throwing up roadblocks against our defense signal.
Okay, let's start with the first roadblock, the viral proteins that stop the signal from even starting.
That falls largely to NSP12. That's the RNA-dependent RNA polymerase, the core replication engine of the virus.
RDRP.
Right. But beyond replication, the sources show NSP12 has another function. It actively inhibits the nuclear translocation of IRF3.
Wait, that's our ignition switch protein, so NSP12 stops the IRF3 switch from ever reaching the nucleus to start transcription.
That's right. It prevents the physical movement required for the defense signal to even initiate. If IRF3 can't get into the nucleus, it can't tell the DNA to make interferons. It's a potent early-stage block.
What about the next roadblock? What if the signal does get initiated?
That's the job of ORF6. So even if some IFN is successfully made and secreted, the neighboring cell needs to receive that signal and turn on its own antiviral state. ORF6 steps in here. It's known to block the nuclear import of STAT1 and STAT2.
And those are the transcription factors that relay the interferon signal inside the cell.
Exactly. So the cell sees the IFN warning, but ORF6 acts like a physical barrier at the door to the nucleus. It stops the signal interpreter, STAT1 and 2, from getting inside to execute the response.
And it does that by messing with a transport protein, NUP98.
It interacts with NUP98. So NSP12 stops the switch from turning on, and ORF6 stops the cell from acting on the warning. It's a very comprehensive two-pronged attack.
And what about NSP1? I remember that one is just a general shutdown mechanism.
NSP1 is the most brutal of them all. It triggers a general host protein translation shutdown. It literally binds to our ribosomes and stops them from working on host mRNA.
So it cuts the main power to the factory.
Precisely. It ensures that even if a message gets through, there are no resources available to act on it. No ability to make new proteins, including more interferons.
This level of understanding, NSP12, ORF6, NSP1, it suggests a deep mechanistic basis for their design choices. They engineered functional suppression because they knew exactly how the virus does it.
And we can see that knowledge being applied experimentally in their research models. This understanding was instrumental in creating their lethal mouse-adapted strain, MASCP6.
Right. Why did they need to create a lethal mouse strain?
Well, mice, especially standard lab strains, have incredibly robust Type I Interferon systems. They're highly effective against coronaviruses.
So when they tested the early virus strains in mice, the mice just fought it off.
They often cleared the infection very quickly. For vaccine testing, you need a model where the virus can actually replicate lethally, so you can properly challenge the efficacy of your vaccine.
So to make a lethal model, they first had to defeat the mouse's innate IFN defenses.
That's it. They forced the virus to evolve mutations like Q493K, specifically to overcome the murine IFN signaling barriers. MASCP6 is applied virology at its height. They use their sophisticated knowledge of IFN antagonism to engineer a model strain.
Which then allowed them to test the efficacy of their functional suppression vaccines.
It's a complete intellectual circle. It shows mastery over viral pathogenesis.
We have navigated some incredibly dense molecular and virological territory. Now, in part 4, let's bring it all back together. Let's synthesize this convergence of pathology for the listener.
The synthesis is crucial. But first, we have to pause and clarify a key distinction in the sources. They mention two types of mimicry, and while they're related to viral proteins, they describe entirely different pathological events.
Okay, what are they?
Functional mimicry and peptide mimicry.
Let's start with functional mimicry. That's from General Zhu's team's work.
Right. Functional mimicry, in this virology context, relates to antibody-dependent enhancement, or ADE. Zhu's team's research suggested that certain non-neutralizing antibodies could bind to the virus and critically functionally mimic the cellular receptor the virus uses for entry.
So they act like a Trojan horse.
Exactly. Instead of neutralizing the virus, the antibody guides it into immune cells through the antibody's own receptor. This was a major virological warning about vaccine safety. It's about how the antibody functions to mimic an entry mechanism.
Okay, now contrast that with peptide mimicry, which is the autoimmunity concern we spent part 2 on.
Peptide mimicry is about structural similarity. This is the theory that small viral protein segments, like RLFRK, look so similar to host proteins like PPIP5K2 or 10NM4 that the immune system gets confused and attacks the host protein.
And the sources are clear that while Zhu's team focused on ADE, the work linking RLFRK to this specific autoimmune risk came from other groups.
Yes, from researchers like Heishua and colleagues, it's an important distinction to make.
It is. 2 separate areas of scientific concern, both stemming from the complexity of this spike protein. So now, the final convergence. Despite using radically different technologies, both platforms arrive at this shared high-risk point, RLFRK and the interferon pathway.
Let's use that comparative framework to summarize it. It lays out the contrast so cleanly. You have 2 distinct problems delivery and 2 distinct solutions, but both result in the same molecular vulnerability.
Okay, let's start with IFN suppression.
For the mRNA platforms, Pfizer and Moderna, the suppression was chemical.
Right.
They achieved it via M1A, a chemical cloaking of the RNA that makes it invisible to sensors.
And for Zhu's platforms, the RGD subunit approach.
The suppression was functional. They used the RGD motif to induce TGF-beta release, which actively downregulates IFN expression. They silenced the alarm after it was triggered.
Different keys, but both successfully lock the same door.
Now for the shared factor, the autoimmunity trigger.
In the mRNA platforms, the trigger was the robust, stable, prolonged expression of the full spike, which contained and shed that exposed RLFRK epitope. And in Zhu's platforms,
It was that deliberate strategy of immunofocusing that magnified the risk by concentrating the entire attack on the receptor binding motif, ensuring a highly specific immune response against that exact mimicry sequence, RLFRK.
and finally the resulting pathology, which is the same regardless of the path taken to get there.
The convergence results in that potential autoimmune attack on PPIP5K2, the kinase essential for making the fuel for the IFN switch.
Leading to that profound risk of biological failure of our innate defense, acquired immunodeficiency.
And simultaneously, that shared risk of neurological demyelination due to the molecular mimicry with 10M4.
What's so fascinating is the sheer elegance of the engineering on both sides versus the inherent biological complexity they ran into. Both approaches were rational, they were cutting edge, highly effective at generating the desired response.
But both introduce vulnerabilities through the exact same critical point of molecular mimicry.
It seems to suggest that when you engineer a protein for maximal immunogenicity and stability, which both the S2P lock and immunofocusing aimed to do, you inevitably expose the most functional, surface-accessible parts of that protein.
And those highly functional surface-accessible parts are perhaps the ones that have evolved to look like crucial host elements, purely by a cosmic coincidence of molecular shapes.
This deep dive has really shown that sophisticated molecular engineering, while it solves the immediate challenge of antigen delivery, it introduces these complex trade-offs. It's all about how the innate immune system is bypassed and how these highly focused sequences, like that critical RLFRK pentapeptide, interact with our own proteins.
It is the ultimate lesson in biological design. Optimizing one parameter, like efficacy, can inadvertently magnify a completely unrelated risk parameter, like autoimmunity, if your optimal target happens to also be a molecular mimicry sequence for a vital host system.
It's the unintended consequence of molecular precision. It really demands reflection. We started this dive talking about the philosophical difference between chemical cloaking and functional antagonism.
And we ended recognizing that both paths led to a shared vulnerability that was just built into the antigen itself.
It raises a profound scientific and, frankly, a public health challenge that future generations of vaccinologists are going to have to address.
Absolutely.
And that leads us to our final provocative thought for you, the learner, to mull over and explore on your own. Reflecting on this shared pathology and the critical role of that RLFRK sequence, how can molecular mimicry sequences be systematically identified and engineered out of future vaccine designs? Or maybe they could be neutralized through subtle amino acid substitutions without compromising the efficacy and stability that you need for a good immune response. Is there a way to stabilize the protein without exposing the mimic?
That question is going to sit at the very intersection of biocomputation and molecular design for years to come.
A truly complex picture of risk convergence. Thank you for joining us on the deep dive.
Always a pleasure to unpack the mechanisms.